So good afternoon, everyone. I'm Beth Dolph with the Office of the Vice President for Health Sciences, and welcome to day 2 of Research Speed Dating Week. We had a great kickoff yesterday, and we're excited to keep conversations going today with a focus on infectious disease. So today we're going to begin with opening remarks from Dr. Nicholas Smith from the School of Pharmacy. After that, we'll do our 1-minute pitches, and then we will have breakout rooms to continue conversation and make research connections. And so I'll pass it over to Jill to introduce Dr. Smith, and thank you all for being here and for your participation.
Well, and a quick thanks to Dr. Dolph for organizing this and bringing us all together with this great opportunity for our speed dating week. It's actually, yesterday was pretty fun, so hopefully you guys get the same out of it today. But more than anything, I wanted to thank Dr. Smith for taking the time to do this. We reached out specifically to the school, Dean Pollack, in the School of Pharmacy when we realized that infectious disease was going to be a topic, because the school has made great strides over the last number of years to pull together top talent from across the country and bring people in to add to their expertise in this particular area. Dr. Smith works alongside a bunch of colleagues who have great expertise in this space, and they thought that he would be a wonderful person to kick us off on the topic. We were just chatting prior. He's been here since 2021. So fairly new to campus, but probably has already figured out the ebbs and flows of the university at Buffalo. But we're hoping that his remarks will set the stage, and that each of you, when you give your talks, that this will stimulate further conversations and more opportunities for collaboration. I just want to mention really quickly that in follow-up at the end of everything that we've executed today, between the pitches and the breakout rooms, our office will help facilitate. We have a survey that we would like you to take at the end. I'll bring everybody back. And that's going to allow us to follow up with each of you who expressed interest in the 3 breakout session topic areas that we've identified to try and connect faculty. So there will be a breakout room on basic science, there will be a breakout room on clinical efforts, and then a third for anybody who's focused in more of the programmatic or community engagement space. And so this won't be all back on you after you have conversations and potentially see opportunities to work across the topics. We're going to take the responsibility for bringing together next sessions and help facilitate those and at least getting them going. So, and with that said, I will pitch it to Dr. Smith to kick us off. But thanks again, Nick, for doing this.
No, thank you, Jill, and thank you, Beth, for organizing this and inviting me. I think these events are incredibly important for research across the campus. But in infectious diseases, just as a byproduct of the field and what we all do and love, I think everyone here is acutely aware that this involves the full gamut of tools, in our kind of clinical and translational science armamentarium. We do deal with a lot of public health questions, a lot of basic science questions, and a lot of clinical questions, that ultimately kind of come back to impacting the patient. So let me share my screen. Perfect. And then, Beth, would you be able to, can you give me a thumbs up if you can see that? Perfect. Thank you. So where I wanted to start is where I think, really what we're trying to all do here is this team science and enhancing the effectiveness of our research programs, with each other and through these collaborations. NIH, I'm sure I don't have to tell anyone here this, but NIH really loves these team science grants where we all come together and collaborate. And one particular phrase in this book, it's available on PubMed, but on enhancing team science that really stuck out to me that was relevant for this session was really that there needs to be this self-organizing teams and groups. And this introduction of newcomers, right? Bringing new ideas to the table, bring new ideas to the team. And events like this really, I think, help do that. Now, where I wanted to focus and just kind of set the tone for a lot of the conversations that we're hopefully all going to have together today is where the preponderance of ideas and funding come from, which is NIH. And so NIAID always puts out the priority areas. And so I just wanted to quickly go through the 5 priority areas that they highlighted and what the Infectious Disease Society of America has kind of responded to these by and highlighting. The first, and I think the most pressing, is advancing foundational research, immune system, host pathogen interactions. The IDSA has approached this as looking at antimicrobial resistance. A lot of people here look at that as well, coming up with new drugs, new drug targets, and finding ideas and ways to collaborate on this. I think is a uniquely impactful and highly fundable area continuing at the NIH. The impact of climate change. This was not specifically mentioned in the NIH, but was specifically mentioned by IDSA, the impact of climate change on antimicrobial resistance. And then the impact of vaccination and infection on reducing downstream healthcare costs. So here we're looking at not just the clinical outcomes, but also the financial outcomes. The second big priority area that I thought would be relevant to our community, our research community, is developing and applying foundational knowledge of microbes in the immune system to develop medical countermeasures. So this includes rapid diagnostics. A big push after COVID, and the example I have here is the use of at-home point-of-care diagnostics being a big push in the infectious disease community and big in research moving forward. Looking at novel therapeutics, which have really, pushed and moved through the landscape in other therapeutic areas, but we really don't see as often in the infectious diseases space. Monoclonal antibodies, immunotherapy, antisense, DNA type molecules, and then phage therapy, for example, the area that my lab works on as well. So developing and moving into these next generation therapeutics and these next generation diagnostics is another big part of the priority. These relate to the non-HIV. They've set aside a third priority exclusively for HIV, dealing with a lot of these same exact questions, coming up with prevention strategies, treatment strategies, and cure strategies. And here I'm showing an example that came out of a really big interdisciplinary collaboration here at UB and at the University of Rochester, University of Pennsylvania, and at NIH. In this case, looking at monoclonal antibodies as a potential therapeutic candidate for HIV and how we can better use these drugs as prevention, treatment, and even as long a way as it might be, even potentially for cure. The fourth area that I think will help drive a lot of our conversations today is looking at asthma, allergic, and immune-mediated diseases and transplantation. This also comes and is motivated a lot of our experiences in COVID. We found that vaccination, of our transplant patient population was an immense and critical a problem that needed to be figured out. This went beyond just developing vaccines and developing therapeutic strategies. Here, for example, looking at the influence of immunosuppressants on whether these vaccination efforts and how we can better use these vaccination efforts. But this is very much and always has been a public health question as well. And so our public health colleagues, I could foresee this being an amazing point of collaboration between us all. And then finally, the kind of the broadest priority is supporting research to prepare and respond to significant biological incidences. And so this could be a pandemic, COVID, this could be AMR expansion in bacteria, this is parasites, this is everything else. This is, for example, the changing demographics of mosquito population. Right now, we look at things like Zika, and Zika happening predominantly in the tropics. But as mosquito populations shift, how can we, as the infectious disease scientists, work together and come up with solutions for the next 30 years, the next 50 years? And so I think this is another big area that both the NIH and the IDSA has highlighted as being areas that we all should begin to think about and to work on. So my little 5-minute intro, I wanted to focus on what I thought were the kind of the big takeaways and the overarching goals. The key is mechanistically novel treatments. So when we're looking at treatment questions, coming up with something that isn't just a me too of a therapeutic class that we might have already had, but coming up with something that these bugs haven't really seen before. And then the other part is patient-centered diagnostics. I was just having a conversation today with Dr. Ray Chaw, who's been doing a lot of HIV stuff. And the biggest cause of HIV transmission today in the US is non-compliance. Has nothing to do with developing good drugs, has everything to do with how we're using them, how we're implementing them, and questions like that as well. Now, that being said, there's still areas for drug development to play a role in those questions, long-acting injectables, for example. And so this really represents an opportunity, I think, I see it as profound collaboration between us all, our natural science colleagues who really inform us on the very basics of the molecules we're trying to create, the biology of the diseases that we're trying to fight. Physics even comes in. I work with some physics trained colleagues on mathematical modeling, things like that. So really, I think in the basic sciences, there's opportunities for collaboration across the board. In the clinical sciences, which I'm guessing is most of this audience, I think we're very acutely aware of clinical pharmacology, clinical microbiology, pathology, and how these influence our decision making. Public health in infectious diseases, I would argue, is uniquely important compared to any other therapeutic area. Public health is important in all of them, but we deal with it so often because of the dynamic and changing landscapes of the diseases that we are constantly working with. One I added in myself here is systems engineering. I am very fortunate that I have a large number of my friends are engineers by training. And the one thing that always strikes me with how they approach problem solving is at this systems level, coming up with the most salient features across multiple domains and trying to integrate them together, which I think opportunities like today really provide us this high level ability to tap different expertise that we all have and how to bring it together in a unified fashion. And then finally, clinical samples, clinical sample collection. We have biobanks that have been generated on campus. For example, I know Dr. Murphy has probably one of the most extensive, or had one of the most extensive H-flu and Moraxella catalogs probably ever assembled. I know our dental colleagues have microbiome data sets. In our lab, we have plenty of isolates on antimicrobial resistance. These clinical samples are the exact tools that we will all use to really perform and realize this bench to bedside and this bedside to bench framework of team science that I know we're all kind of acutely after. And so for me, kind of just trying to set the stage for hopefully some fun discussions today, I just wanted to point out, where NIH is at, where the funding landscape is at, and where each of us can potentially integrate our tools and our skills into our collaborations, into our newfound teams. So with that, I thank you for letting me kind of introduce the topic and participate. Thank you, Jill and Beth, and happy team building to everyone here today. Hopefully we come up with some great ideas and some great projects.
I love that you used that book as a reference. I
Thank you.
spent
Yeah.
an hour with that.
Yeah. Well, it was, especially I think the other thing that's coming up that I see a lot in the funding is, the center grants and center funding seems to be very much, a big fan, and team science is an acute part of that as well. So, yeah, big advocate.
Fun stuff. Does anybody have any questions before Dr. Dahl kicks us off into the quick pushes? Does anybody have any questions or comments for Dr. Smith? Okay, Dr. Dahl, speed dating, research dating.
All right. Let's see. Our first pitch will go to Dr. Paz.
Hello, everyone. My name is Aviv. I'm a member of the Structural Biology Department, and I'm housed at the Hoffman Woodward Institute. I mainly work on human membrane transporters, and I just started branching into multi-drug resistance efflux pumps that banish many therapeutics outside of resistant bacteria. So in the last few weeks, we started working on trying to solve the structure of the tuberculosis TEP protein that is related to a multi-drug resistance of many therapeutics that are used for the treatment of tuberculosis. I don't have to say to this crowd that tuberculosis has tens of different resistance mechanisms. This transporter isn't the main one, but still, if you could find some weaknesses in an organism, why not try and exploit it? So my aim is to solve the structure with and without drugs, and then this could open, a new window for and many other things. Another protein that I just got the DNA for is the Acinetobacter baumannii ACE1 chlorhexidine efflux pump. That's a really interesting transporter because there are no solved structures of this transporter or of any homologs, and it's predicted to be a novel fold that is only used in bacteria. So this could be a really great target, and this transporter is important for the resistance of the bug against chlorhexidine, and other homologs are important for the resistance against other bactericides as well. So that's something that is super interesting, and it could be very promising. Was it a minute? Maybe.
Yes, we'll say yes. It was close enough. Thank you so much. We'll go to Dr. Baker.
Hi, everyone. I'm Kelly Baker. I am Associate Professor in the Department of Epidemiology and Environmental Health in the School of Public Health and Health Professions and Director of the Center for Climate Change and Health Equity. I don't have a specific pitch today because I'm relatively new to UB, and so part of my purpose in being here is to find areas of mutual interest and build ideas from that. I have 20 years of experience in global health infectious disease epidemiology. Most of my work is very translational in nature. It's about unpacking the mechanisms of how disease is being transmitted in communities and identifying and evaluating interventions to prevent that. So, for example, some of my projects include collaboration with the Carter Center and the government of Ethiopia on guinea worm eradication, working with the government of Kenya, Ministry of Health and State Department of Urban Planning on enteric disease transmission, diarrhea, malnutrition in kids, really thinking about community development and how community development can provide barriers that stop those pathogens from circulating in communities. I've had several grants from NIH and the Department of Defense as well for the development of rapid diagnostics and surveillance tools for enteric pathogens, including antimicrobial resistant enteric pathogens. I also do have biobanks here with thousands of specimens that have been collected through these community-based sort of surveillance studies of human, animals, and environment. So we have a lot of different interesting resources in our biobank that come from a lot of other places around the world. And then I guess last, I just want to give sort of a pitch for the center. You probably are going to hear from someone else here on the call. Mark Janko is also one of my people here at the center. We do have a current pilot grant call out for people that are interested in participating in future climate and health center grant applications. We're looking to build out a community that's working in that space to be competitive for those center grants. I think I've probably gone over a minute, but I look forward to talking to you all again in the breakouts.
Thank you so much. And we'll go to Dr. Kumar.
Hello, everyone. So I am Dr. Vitash Kumar. I'm from Oral Biology Department, School of Dental Medicine. So what I've been working since my postdoc and my faculty years for the last 2 years on oral fungal infections, and particularly with Candida albicans. And so my lab works in two parts. One part is basic science, basic versus translation science, where we work on how the host pathogen interacts with each other for disease progression in monospecies and multi-species oral infections, including bacteria and fungal interactions. And the other part is where we are trying to target antifungal resistance using antimicrobial peptides, especially present in human saliva. And one such molecule we have been working since like 10, 20, 12 years is histatin 5. It's a small peptide with like 13, 14 amino acids, but it's got a very potent antimicrobial activity. And especially in these like last 3, 4 years, what we have been working on that when you mix with salivary metals such as iron and zinc, it increases its potency. So it becomes much more effective as compared to the peptide itself. And it has been effective against not only albicans, Candida albicans, which has been effective against multiple fungal strains, which are found in oral cavity. And we have been testing that in also where they are effective against those oral bacteria that could be either commensal or maybe pathogenic, depending on the situation of the host. I want to let's collaborate if somebody is interested into looking into a polymicrobial drug resistant phenomena using these antimicrobial peptides and zinc together. And I'm also ready to talk to more about like if somebody is much more interested in basic mechanisms of oral fungal infections versus any other microbes together in oral cavity or in gut probably. So yeah, so I'm much more interested in talking more about it.
Thank you so much. Dr. Bloom.
Hi, my name is Amanda Bloom, and I'm with the Department of Microbiology and Immunology. And I've worked for many years with Dr. John Panapinto, and now I'm kind of venturing off on my own as a new assistant professor. And we have found that with the pathogenic fungus that we study, Cryptococcus neoformans, which predominantly affects individuals who have HIV, it's very susceptible to proton pump inhibitors, the common proton pump inhibitors that about 25% of all adults are taking. And not only is Cryptococcus super susceptible to them, but we've also found that the other human pathogen, Aspergillus, is also very susceptible, while some other fungal pathogens are not at all. And so we're looking into trying to figure out this mechanism by which it is susceptible. And we have preliminary evidence suggesting that the mitochondria is severely dysfunctional in the presence of these proton pump inhibitors. We've also done some preliminary work with Dr. Zachary Falls in medical informatics, and we've seen that there's about a 50% reduction in solid organ transplant recipients to have a cryptococcal infection if they're on proton pump inhibitors. So we're kind of looking more into getting some more medical informatics type of data, as well as we are looking at the benchwork side of things, looking at targets of these PPIs in Cryptococcus. And I guess the main reason that I'm here is this is very new territory for us. We're like jumping over a bridge and getting into the drug discovery, pharmacokinetics types of study sections. And like I said, this is novel for us, and we're looking for advice. We're looking for people who might want to collaborate, looking into the mechanism more with us, as well as the informatics side, to kind of bring this into more of a clinical realm as well.
Thank you, Dr. Bloom. Dr. Vassaretti.
Yeah. My name is Purna Vasireddy. I'm a postdoc in Dr. Martinovich lab. I'm a trained synthetic chemist. Our lab's primary focus is my boss, Dr. Martinovich, is in structural biologist, and his main focus is on developing methods for electron microscope, which is called our special topic is microcrystal electron diffraction. In addition to that, my major focus in this lab is we work on small class of compounds called peptoids, which are peptomimetics, class of peptomimetics. And these class of compounds are shown activity as an antimicrobial and anti-cancer and various other diseases as well. And we started our work as like taking the known compounds to solve their structures, because so far there were no structures were solved. Crystal structures were solved for this class of compounds. We started from there, and our future direction is towards like developing a class of peptide compounds towards antimicrobial and anti-cancer related diseases. Yep, this is my.
Thank you. Dr. Joseph Crane.
Hello, everyone. I'm actually not a doctor yet. I'm a DMP student and also a research assistant. So I've been working with two separate mentors. One whose primary research is on climate change and how environmental factors affect chronic disease, and the other is more so on HIV research and how it affects HIV positive mothers. My personal research that I'm working on currently is a systematic review on how climate change impacts access to care and infectious disease for people who use drugs. And another area that I've been working on is provider readiness locally and in the United States for tropical diseases as in a warming environment, and how prepared we are to address these concerns as they come to other nations that have not experienced them before. I also want to mention that I've done some global traveling recently. I've been to Ghana, and I'll be going to the Dominican to do some HIV research there. And I plan to incorporate kind of a global view, but also using it to inform things for right here at home as well. And I really appreciate being able to be involved in this and being among so many established professionals. So thank you very much.
Thank you. That was everyone I had who had signed up. Did I miss anybody who wanted to jump in?
I can jump in real quick just because Kelly mentioned me. So I'm Mark Janko. I'm an assistant professor at the Center for Climate Change and Health Equity in the Department of Epidemiology and Environmental Health. I'm actually a geographer and a statistician by training. And so I think of myself as a statistician who does field work. And I've spent the past 10 to 12 years working mostly on vector-borne diseases in the Amazon and Congo basins. I currently have an NIH grant that takes place in the Amazon, and it's studying how climate and environmental variability shape human mobility and how that in turn shapes parasite population structure and parasite diffusion of malaria parasites. I have similar projects underway on other infectious diseases in Madagascar, and I've done a bunch of work in the Congo where I did my PhD several years ago, about 10 years ago now. So that's kind of me in a nutshell.
Thank you. Oh, yes, go ahead, Dr. Das.
Yeah, my name is Bhaskar Das. I'm professor in the School of Pharmacy and Pharmaceutical Science, as well as adjunct professor in the pharmacology, toxicology at the medical schools. And it is just 1 year I finished here in the UB. I came from Mount Sinai School of Medicine. So I am the chair and also division chief of the drug discovery and biotherapeutics. So my background is a chemical biology. So basically we develop the small molecules for the infectious diseases. My research focus on the neuromicrobiology, how the microbiomes modulates the memory. And also another area I am working with exposome, how the environment pollutes, that affects the memory development. So I have around 62 patents for drugs in the infectious disease area approved, FDA approved from my lab, the crisaborole and travaboroles, the two drugs I developed from my lab. And it is basically anti-infectious diseases. So I am keen interested how to develop here at UB the group, the neuromicrobiology areas and the micro to memory. So I have around 6,000 compounds. Majority are boron-based compounds. If someone interested, they have a specific targets, so I can develop the compounds, or I can send the list of my existing target to it and hit to lead compounds if someone is working, and I can share the compounds with them. So here my intention is to understand which other faculties as I am new to here, and I want to expose with the neuromicrobiology area at UB. I'll stop it here.
Thank you. Was there anyone else who wanted to do a pitch? I think we covered everybody. Okay.
So I'll open the breakout rooms. We don't drive. It's all up to you guys to just have a chance to meet each other and speak more on the topic. You can come and go from the breakout rooms. Some of the suggestions that I would have though, if you've seen a recent funding opportunity or you've seen something in the past or you know something that comes up, maybe bring that to the forefront of the conversation on ways that you could put a group together to potentially collaborate. I would keep in the back of your mind that UB does invest in centers and institutes when we have a core group of investigators interested in critical topics. So that's always something to think about. And then I would also say that we do occasionally out of our office run and pitch out seed funding opportunities. So if a group of you come together and recognize that there is a topic that you can converge on, that could lead to application for another funding external mechanism, but you need some seed fundings to collect data and get things going. That's always something that you could talk about too in some of the sessions besides your work. So we want this to be productive for you. I think we'll probably give you about 20 minutes and then call everybody back. I've figured out how that can interrupt breakout rooms, which is actually quite fun. And I'll send a message and then bring everybody back and then we can push out the poll and see who wants to continue conversations further. Does that work? So you should have a chance to opt in to any of them right now. If you need to, you'll see breakout rooms in the bottom right-hand side of your menu ribbon at the bottom of your screen. Well, thanks to all of you for coming back. We hope that the session went well. As I mentioned at the beginning, we're going to launch a quick poll, and the whole purpose behind it. There are no open-ended questions. It's just to basically ask you if you're interested in follow-up, and hopefully that you've had an opportunity to learn about somebody else's research program that you might have been unfamiliar with. And when I download the results and the data, you'll see an email from me tomorrow with a colleague in the Office of the Vice President for Health Sciences, or Office of the Dean in the med school, well, both probably, who's going to help us create follow-up sessions. So if you say yes, then you're probably going to get a rally poll. I think what we're going to do based on timing and everybody's availability, we'll aim to get those sessions started in sometime in early March. Any thoughts, any comments? Dr. Smith, do you want to say anything as we're closing out?
I don't know about anyone else, but I just want to thank you again, Jill and Beth for organizing this. But I kind of actually upset I didn't get to any of the other sessions. We had a great conversation, but all of the rest of you, I was actually very interested in chatting as well. The 30-second warning came up so quick, but it was really nice to hear everyone in their pitch today, and I'm looking forward to hopefully catching up, and I'm sure I'll be reaching out to some of you that I didn't get to talk to and some of you that I did as well. So, but thanks again for coming, and thanks for having me.
I second the Dr. Dolph thing. I say to my team all the time, I feel so lucky to work with such smart people. So, and talented and committed. So anybody else have any other thoughts? Thank you. I see great responses for the survey, which is extremely helpful. It'll help us follow up with you. The last thing that we want to do, we talk about this all the time. The last thing that we want to do is bring everybody together. Dr. Brashear, I kid you not, if any of you have been in her office or in the room that we hold cabinet in, the conference table, she literally has these bright red buttons on the table that says easy button, and you can hit it and it dings or it go. And the whole idea is that we want to make it as easy for all of you as possible in order to advance your research programs. And that's what an administrative office, and that's what drives all of our efforts and those of my colleagues across the screen. So we just don't want to bring you all together and say, hey, how was it? And then not follow up and help facilitate additional efforts. So I will tell you that I'm probably going to engage the CTSI for any of you that had topics that you were interested in as related to clinical efforts. I'm going to loop in Dr. Sathi Kachin-Noyes because collaboration team science is part of their mission as well. So we'll broaden the conversation and see what resources we can put behind all of this. So